Hello everyone. In today's class, we will discuss about salt-gel hypothesis. Okay. See, in the earlier class, I have discussed about pseudopodia. So, what is pseudopodia? Pseudopodia means it is a false feet. Why it is called a false feet? Because it originates or it is formed when there is a requirement. Okay. And once the requirement is done, then the pseudopodia degenerates or it disappears. Okay. Now in this class we will discuss about this formation of pseudopodia through salt-gel hypothesis. Okay. Now, pseudopodia is formed in amoeba. Okay. So suppose this is an amoeba and this is a food, okay, located near an amoeba. Now what amoeba will do, it will extend or form the pseudopodia. Okay. So suppose this is again in the next stage, the pseudopodia will be formed like this. Okay. So it will extend towards the food to capture the food. So now once we study the pseudopodia area, once we study this area, suppose if we study this area, we will see like this, that the formation of pseudopodia takes place. Okay. So this, how this takes place? It takes place through cytoplasmic streaming. Remember the term cytoplasmic streaming. Now how and why this takes place? Okay. Suppose this is the pseudopodia, this is the extended part. Okay. Now what happens is that the outer portion of the pseudopodia, this is the ectoplasm. Okay. And the inner portion is the endoplasm. Okay. Now what happens is that the ectoplasm is gelatinous in nature, while the endoplasm is fluid in nature. Okay. The endoplasm is more fluid in nature. Okay. Ectoplasm is gelatinous in nature and endoplasm is fluid in nature. Okay. So this is known as gel and this is known as sol. Okay. Now you get how the term came. Now what happens is that the cytoplasm moves from the endoplasm to the ectoplasm when there is a stimuli. Suppose in this case there is a food. So what will happen is that the cytoplasm will move from the endoplasm to the ectoplasm. Now in this next stage what will happen is that the cytoplasm will move like this from the endoplasm to the ectoplasm. Okay. Now we will say, sir, why this takes place, this type of movement? There must be some reason for this type of movement to take place. Yes, there is a reason and this is the most important portion of this discussion. Okay. Now see, listen carefully. Okay. What happens is that in the ectoplasm there is presence of actin. Now actin is a type of protein. So what happens is that in the normal state, when there is no stimuli, the actin is like this, means it is polymerized form, in the form of a polymer. Okay. Now what happens when there is a stimuli, the actin will depolymerize. Okay. The actin will depolymerize like this, means it will break down. So what happens when the actin will break down in the front portion? The osmotic pressure changes. Okay. Now once the osmotic pressure changes, the cytoplasm from the inner endoplasm will move towards the outer ectoplasm. Understood? I hope this much is clear. Now this, once the cytoplasm moves from the endoplasm to the ectoplasm, then again in the next portion repolymerization takes place. Now how this takes place? Look carefully, okay? Suppose this is the extended part now. Again the actin will repolymerize, okay? Now in this, why this repolymerization takes place? Because it forms the firm structure so that the pseudopodia will not degenerate easily, or as long as the stimuli is present, the pseudopodia is present, okay? So you see this again repolymerization. Here you have seen depolymerization, here you will see again repolymerization, okay? Now the cytoplasm is like this. So the pseudopodia has extended or has been formed, okay? Now once the stimuli is absent, or once the stimuli is gone, what will happen? Then again this process will be like this. This actin will again depolymerize, and the cytoplasm will go to its original position, means retreat. And once it will retreat, the actin will again repolymerize to form this final stage, okay? The actin will repolymerize and form this final stage, okay? I hope you have understood this cycle. First the actin is polymerized form, then it depolymerizes, then again it repolymerizes, okay? Now this cycle continues. Again it will come back to its original state once the stimuli is gone, and again repolymerization will take place, okay? So this process continues as long as the stimuli is present, and when the stimuli is gone, then the process is again taken place to make the pseudopodia to come back to its original state, okay? I hope this much is clear. So thank you everyone.